reproductive season should best explain plasticity in the full model. Now on to my experimental crossing study, where I will answer the last three questions. So we'll start with our nuclear or cytoplasmic genes. My two hypotheses are that first I'll find evidence of cytoplasmic inheritance, or I will not. And to do this, I'm taking advantage of that genetic variability that we saw in the populations of Plantago. So if you remember, individuals down here from the south have very low values of plasticity, whereas individuals from up here in the north have high plasticity values. So I selected the individuals with the most extremes that I could find in our populations. And I'm using those as parents in an experimental cross. In this diagram, I'm showing cytoplasmic genes in the outer circle and nuclear genes in the inner circle. So for this cross, this low plasticity southern parent is the seed parent, and this high plasticity northern parent donated pollen. So the F1 individual has the southern cytoplasm and 50% of its nuclear DNA from each parent. I'll also complete the reciprocal cross. So I have these two reciprocal full sibling populations to compare. Extend that out a little bit more. I decided to cross two of those because Plantago lanceolata is self-incompatible. I needed independent individuals in order to cross to make an F2 family. And in each of the F2s, the parental alleles are expected to segregate out in the nucleus, because at any one locus, it could be homozygous for the high plasticity allele, homozygous for the low plasticity allele, or heterozygous. All individuals from the population will then be cloned into 4 replicates, and 2 clones per genotype will be placed in a cool environment under 15 degrees Celsius days, 10 degrees Celsius nights, with the other 2 clones placed in a warm environment of 27 degrees Celsius days and 20 degrees Celsius nights. And then I'll measure thermoplasticity of many different traits as the difference between that trait value in warm and cool environments. I'm interested in studying the plasticities of these reproductive traits. First, days to flower, because that can influence the environment in which those flowers and seeds develop. Second, spike reflectance, because we know that influences thermoregulation. Third, spike length, because that's a proxy for the number of flowers on that spike and thus potential clutch size. Third, trichome density, which are these small microscopic hairs on the flower. And these appear to be lightly colored under the microscope, so they may influence reflectance. And because they're hairs, they may also influence the boundary layer, and therefore they could influence thermoregulation. The length of these scapes, because having a long scape may allow individuals to dissipate heat more readily, and having a short scape may keep them closer to the boundary layer of the ground and observe more thermal radiation from the ground. And the percentage of viable pollen, because that's a proxy for male fitness in this wind-pollinated herb. I'm also interested in measuring several vegetative traits, including the diameter and height of these rosettes, the length, width, and specific leaf area of the longest leaf, as well as its fresh and dry mass. Because, as I discussed, body size is important to thermoregulation, body size and shape. And these are all proxies for that measure. So, to address my 2 hypotheses that plasticity is inherited through cytoplasmic or non-cytoplasmic genes, what I'll do is look for phenotypic differences between full siblings in these reciprocal populations. And I'll do this using t-tests and ANOVA. So this is where I am currently in my crossing experiment. I have gone through and crossed independent parents to create 7 different F1 reciprocal families. I went through and did this because we weren't sure which reciprocal families would be compatible with each other because Plantago is self-incompatible. Right now, I'm in the process of producing these F2 individuals using controlled crosses. One thing that I found between my 7 reciprocal F1 families is that in every single case, when the seed parent was from the north, a high plasticity northern parent, it produced larger seeds than the reciprocal, a low plasticity southern parent. My third question, what is the genetic architecture of phenotypic plasticity? I'll test these 3 hypotheses, that it supports the overdominance model, the allelic sensitivity model, and the epistasis model. And to test these hypotheses, I'll first identify markers across the genome that are the same markers in the parents, the F1s, and the F2 individuals. I'll then order those markers onto a recombination map and conduct QTL mapping to associate genomic regions with the phenotype. So first, to identify those genetic markers, extract the DNA from the individuals, and I'll digest it with 2 restriction enzymes. That will produce a fragment of DNA that's unknown, but has a restriction cut site from enzyme 1 on one side and enzyme 2 on the other side. I'll then anneal adapters that are specific to those restriction enzyme cuts. Here I'm showing EcoR1 and MSE1 as an example. And these adapters include an individual barcode so I can identify this unknown fragment to a given individual. I will then use these PCR primers to attach Illumina flow cell annealing sequences as well as a multiplex barcode. And that will produce final sequencing libraries that are like this. And it will allow me to pool multiple individuals into the same sample, send it off for Illumina sequencing, and by demultiplexing the multiplex and the individual barcode, I'll be able to then identify which individual that fragment DNA belongs to. So after preparation, I'll send off those libraries to Illumina for sequencing. I'll then demultiplex the sequences and identify markers that are traceable from the parents through to the offspring. I'll then conduct recombination mapping to order markers on a genetic map. And to do this, I'll use the recombination frequency, which increases between any 2 loci as the distance between those loci increases. I'll place those onto a genetic map using the map unit centimorgan, which is simply the recombination frequency times 100. So for example, if I have two markers and recombination was observed 15% of the time between those markers, I'll place those on the map at 15 centimorgans apart. Then to get the order, we simply add in a 3rd marker. So if marker 3 is 25 centimorgans from marker 1, it could either lie 25 centimorgans in this direction or 25 centimorgans in this direction. But since I know its relationship to marker 2 as well, then it must go there on the genetic map. Once I have a genetic map, I will conduct QTL mapping, in which the family that I create will be used to associate the genotype with the phenotype. And so here I have my low plasticity parents and my high plasticity parent trait values. All of the F1s have 50% of each parent's DNA in their nucleus, so they're expected to have a trait value somewhere in the middle of the 2 parents. The F2 should produce a longer tail distribution because some individuals will have more of parent 1's DNA and other individuals will have more of parent 2's DNA, with many individuals in the middle. I'll then scan the data for evidence of QTLs, and this is an example result that I made using Arabidopsis lyrata data. Here I have logarithm of the odds score on the y-axis.